the modern synthesis was incomplete. Evolution of biological organization depends on development. Sean B. Carroll, in his Endless Forms Most Beautiful, notes two classically Darwinian concepts in Julian Huxley's Evolution, the Modern Synthesis. First, that gradual evolution can be explained by small genetic changes that produce variation, which is acted upon by natural selection. Second, that evolution at higher taxonomic levels and of greater magnitude can be explained by these same gradual evolutionary processes, sustained over longer periods. The modern synthesis established much of the foundation for how evolutionary biology has been discussed and taught for the past 60 years, up to 2006. Then Carroll succinctly states the problem with this perspective. However, despite the monikers of modern and synthesis, it was incomplete. At the time of its formulation and until recently, we could say that forms do change and that natural selection is a force, but we could say nothing about how forms change. Mutations have non-random features. As will be discussed in more detail in Chapter 16, a variety of mechanisms generate non-random changes in genomic DNA sequence elements. If genetic mutations are defined as changes in DNA sequences that are passed on to future generations, then mutations are not simply random changes. More importantly, mutations such as gene duplication events, which are quite common in molecular evolution, create duplicates of genetic elements that have already been subjected to natural selection and have already been proven useful in previous contexts. These duplicates, therefore, have significant non-random aspects. Duplicated genes can and do change by means of classical natural selection and generate new structural and functional capabilities, but they begin their evolutionary history in a non-random fashion. The modern synthesis perspective on mutations as purely random events must be tempered with a broader 21st century perspective that recognizes such non-random aspects. Reuse of useful genomic sequence elements implies conservation and/or duplication of those elements rather than creation from scratch. The deep homology of genes that determine major features of animal shape, form, and capabilities. In Endless Forms Most Beautiful, Carroll provides an accessible account of some of the major new principles that Evo Devo has added to evolutionary theory. Contrary to the expectations of any biologist, most of the genes first identified as governing major aspects of fruit fly body organization were found to have exact counterparts that did the same thing in most animals, including ourselves. This was not expected because biologists had assumed that different animals would be genetically constructed in different ways. In the heyday of the modern synthesis, selection was seen as the sole creative force in natural selection, and variation was assumed to be random, as discussed earlier in this chapter. If mutations are random and selection is the creative force, then separate gene pools would be expected to evolve according to separate ecological selective pressures, but the ways in which they would change, the particular set of accumulated random mutations that take place, should have separate histories. Continuing with some highlights from Endless Forms Most Beautiful, Sean Carroll points out that the development of various body parts, such as eyes, limbs, and hearts, vastly different in structure among animals and long thought to have evolved in entirely different ways, was also governed by the same genes in different animals. Highly conserved genetic elements that determine development are examples of what is often referred to as deep homology, an important new perspective for evolutionary theory. Modularity. Another important principle highlighted in Endless Forms Most Beautiful is that animals have modular architecture. They are constructed from metaphorical building blocks. A basic theme of animal design becomes obvious when one tries to figure out just what bone or tooth one has found in that shovelful of Florida river gravel. The challenge of the game is both to match the fossil to a species and also to determine where in the animal it belonged. Why is this so hard? This is one demonstration of a basic fact of animal design. Related animals, such as vertebrates, are made up of very similar parts. Other questions addressed by Evo Devo. Some of the other major questions that can now be addressed empirically include answers to the following questions as summarized by Carroll. 1. What are some of the major rules for generating animal form? 2. How is the species-specific information for building a particular animal encoded? 3. How does diversity evolve? 4. What explains large-scale trends in evolution, such as the change in number and function of repeated parts? In addition to deep homology, another important general answer can be found in our newly acquired understanding of regulatory elements such as Hox genes and transcription factors, which are briefly discussed in Rethinking Evolution.